So what I've done is I've kind of adapted an old talk, which I think none of you have seen, so I think should be fine. Taken out a chunk and put in some of the new interesting results that are coming in, and hopefully I get time to get onto that. But I do want to encourage you, this talk I've mainly given for neuroscience audiences. So I've adapted it somewhat, put in more equations for the math audience, but please at any time do stop me and ask questions. The goal here is for you to learn as much as you can about a topic, which, well, I see a lot of Eve's lab here, but other than those, a lot of you may not know too much about. So if I skip things or, you want me to focus on one thing, just please ask at any time. Okay, so this is work with Jonathan Cannon, who I was very lucky to receive. He actually applied to Eve to do a postdoc, and she nicely passed him on to me. He had come from Nancy Capel's lab as a PhD student. He's now got his own lab at McMaster in Canada. I should know where in Canada. I think Waterloo or somewhere, but I don't know where that is. Sorry, I apologize to the Canadians in the online audience. Okay. So now my screen is not accepting a move forward, which has never happened before. I can go to here, maybe. No. Huh? Oh, okay. It was on there's too many screens. Okay. So here's a brief outline. There'll be some introduction about homeostasis in neurons. First, I hope you all understand what homeostasis is. The classic example is body temperature, or as we approach winter, keeping the buildings the right temperature. Things get too cold, you want something to heat them up. If things get too hot, you want to cool them down. Our bodies do that. And there are numerous mechanisms for different features that are essential for life in our bodies. Partly what I'm going to get at is things are a little bit different with neurons, but homeostasis is important. I'll point out a stability problem, how we can solve it, why you might want more than one control systems. And we kind of had left it at there, but there's some recent evidence that there are more than one control system. And I'll explain, how you can distinguish one system with multiple outputs from maybe two different systems. Okay, so here we go. So, just for those of you who, would like a bit of neuroscience background, on the left, on the x-axis is time, on the y-axis are just different example neurons. Black dots are the spikes, and hopefully you just get an idea. Things are variable. Okay, I think this was visual cortex. Yeah, V1, and this is spontaneous activity, which means probably in the dark. I don't know, the animal's effectively dreaming or something, but there's basically, without any change in input, even neural activity in a circuit is just jumping around. I kind of think of this as jumping through different attractor states. But the key is, this is unlike any other homeostatic system I know of, because with our body temperature, we try and keep it, if you're in America, at 98.6, if you're everywhere else in the world, 37. But you don't want it jumping 0 to 100, right? We would be dead. But with neurons, you do, because their goal is not just to maintain constant activity. There'd be a waste of space. The whole goal is to process information, which requires variability. And the way I think about it is, and I should say I'll be jumping through different types of models. The simplest model is based on this curve, firing rate model, where just as a function of if a neuron is getting more input, then it's going to fire more spikes per second. So it's kind of like a gross feature model that kind of works if you're averaging over many cells with without too rapid variations or synchronizations in spikes. And the idea is there's some sort of threshold, which is, you talk about a threshold current, but here often in theory, we talk about like the half max of the firing rate curve as a kind of threshold. And you also have some inputs. And the inputs are also controlled by a neuron because they depend on the conductances of channels. And what a neuron is essentially trying to do, rather than just hit one fixed point of firing rate, is trying to control its range of outputs and match it to whatever inputs it's getting. Okay? And right away, as soon as I say a range, range has a minimum and a maximum, or a medium and a half width, or so on. you've really got two parameters. So it might give you an idea that it might be actually good for something that is varying across a range to have more than one control system, because one control system can only control one variable, essentially. Please stop me if you... So, the very first data on homeostasis, actually, the earliest was probably done in Eve's lab on a kind of motor system. The earliest data is showing two mechanisms I know of is from Gina's lab, also at Brandeis. And what they showed is that this is in the visual system. And one of the nice things, because we have two eyes and the right bits of cortex, you can basically do things with one eye and not the other. So you've got an internal control within the animal. And you look at neurons that receive input from one eye. If one of them is basically, in this case, there's an eye patch, which, at the level of when you start looking at the cortical activity is not as clear as stopping inputs, because activity can rise a couple of steps along the way. But you basically change the inputs and certainly reduce them or reduce correlations in them that you get from normal visual activity. And what happens is if the inputs are reduced, then the neuron responds so that it scales up the channels, the input channels or the input conductance. So that means for any spike on a presynaptic cell, any spike arriving at that neuron, you're going to get more current coming to the neuron than you would otherwise. So this is homeostatic. You reduce the inputs, and it's kind of like turning up the gain on the amplifier coming in or something. Each input now is going to give it more current. Okay. But the other thing that happens, if you say, well, as a function of the current injected to that cell, how easy is it to get the cell to spike? You also find a shift in threshold. So now you need less current to generate a spike in the cell. So these are kind of two distinct properties. The one on the right, the shift in firing rate response to current is FI curve, is called intrinsic homeostasis. It's kind of like intrinsic properties, the channels, often potassium and sodium measured in the cell body. And this is called synaptic scaling. And these are the connections between cells getting stronger. Okay, so they're two distinct things. They all depend on channels at some point, but very different channels. And so you can think of it this way, that you do some sort of sensory deprivation. The excitatory inputs get stronger with their connections coming in. Inhibitory inputs, which I'm not going to look at so much, also weaken at the same time. But then the cell can make it easier to spike by having more sodium channels and fewer potassium channels. And both those have been seen. Okay. So really, even in this figure, there could be four mechanisms, because you could have potassium channels, sodium channels, excitatory, AMPA conductances, inhibitory GABA conductances, all changing. They're all different proteins, so they could be controlled differently or together. So I'm going to kind of skip. So homeostasis is really classic. We are getting an engineering major now. I assume they do control theory and maybe first, second year. It's kind of classic undergraduate engineering. And while there are different types of controller, the one that's most robust is integral feedback control. So this is kind of the block diagram, the way the engineers look at this. I'll show you the equations underneath. But the idea is, some system has external input. There's some way of changing the gain to that input. Then the system has a response. Here would be the firing rate of a neuron. And there's some target response, okay? And that target, and you compare the response with the target, you subtract the two to get an error signal. Then you feed that error signal back in, and the way it's fed in this type of controller is through integration. Okay, so this can then be seen, the math equations are kind of probably easier for us all to understand. We've got the firing rate of a cell. It's some function of its inputs coming in at a rate. The input conductance, which is basically what I said, scaling the inputs. And here I just have minus the threshold in this simple model. So if you increase the threshold, even given the same inputs, it's harder to spike. And what the controller is doing in this case is just controlling the gain And because you have an integral controller, the rate of change of this gain signal is simply proportional to the error. And then you could ask, well, what is the fixed point of the system? Well, fixed point is where rate of change is 0, which just means the error has to be 0. And then you just have one, when you have one controller, the only constraint is the sign, right? You better send in the error. You better put the heating on when it's cold and the cooling on when it's hot, rather than vice versa. That's the only constraint. So these can be very stable and fairly simple to engineer. I'm going to take a little bit of, do a little bit of, anytime you do biology and you're not, this is not your area of expertise, only measuring this molecule in this experiment, it's a huge simplification, right? But even the simplified model looks, kind of complicated, but the idea is, how could this be operating in a cell? You have neurons producing spikes. The spikes are spikes of voltage. But when the voltage goes up high, for example, calcium will enter the cell body because there are voltage sensitive calcium channels. And that is already, I kind of think of that as a filter of the voltage. The channels aren't exactly synchronized. They've got a time constant. That can lead to activation of kinases. So this is a classic one, which they know is important in this, kinase 4, which is a transcription factor. Transcription, these are the stages where we don't know precisely how exactly changes in input are changing the calcium and changing which proteins essentially are going to be produced in what extent of rapidity and so on. But there's transcription. I mean, the net process is clear. There's transcription, there's a change in the amount of mRNA. And when you have different amounts of mRNA, you have different amounts of protein. And then those proteins are going to be transported and inserted into the cell membrane. And then when you've got new proteins in the cell membrane, these are channels that can let ions in and out more easily. So you've got a change in the electrical properties, okay? Or these could be proteins that are synaptic proteins that make the connection stronger or weaker, okay? So, this whole process can happen, and, different ways of looking at it. You've got firing rate of cells, opens calcium channels, activates kinases, and there's transcription leading to translation, membrane insertion via mRNA. And we have kind of simulated this, and this is kind of based on Tim O'Leary's framework when he did this in the Marder lab. The key is that each of these processes are they're essentially chemical reactions. The reaction rate will depend on the amount of the reactants. Then there's degradation. When it's a kinase, the degradation is really dephosphorylation. And if you look back at the engineering framework to get to get the control is really this level where you have the integrator. That is what is setting the fixed point and deciding if you have any further change in the system or no. So one question is, well, how do you get an integrator out of this? And the basic way is that if we go through these equations, we got all these rates of change that depend on the previous step, which is producing the reactants. If the loss of one of these is basically independent of the amount, then you can have an integrator. So if, for example, the dephosphorylation of the kinase, if that is independent of the concentration of kinase, then the kinase is just integrating up the amount of calcium it gets. Now, this may seem impossible, but if you've done biochemistry, I'm not sure which class is taught, but there's Michaelis-Menten kinetics. And this basically is the idea that when, most, I should be careful saying most, but biological reactions require catalysts, right? It's not just they always occur. If the rate is constrained by the amount of catalyst rather than the amount of substrate, then you can have more substrate, but you're not going to get a faster rate. Okay, and certainly that can happen in dephosphorylation. It could happen down here that if nothing really is stopping channels being inserted along those ways, along those pathways, but removing channels is limited by the amount of ubiquitin or something, so that you could have double the number of potassium channels. They're still going to be removed at the old rate from the cell. Then insertion in the cell membrane will be an integrator. Okay. And we did some models at this level where you could say, well, suppose this is the concentration independent step, and we put in Michaelis-Menten basically for one of those, or this is the Michaelis-Menten step. And essentially, all these pathways, they're depending on the firing rate, the spiking and calcium influx at some point. And the question is where, does the integration step occur after those pathways have diverged or before they diverge? So in this case, you'd have one controller, which is very easy to make it stable with different outputs. So you'd say, you'd have a protocol, change the amount of calcium coming in. Now you're going to change both inputs, synaptic conductances, and the firing rate of the cell through the FI curve. If it's this case, then you've really got two controllers. And this is where we can look at that and how that can happen. So going back to this engineering framework, this is what I showed you. With one controller, you have one error signal. And essentially, what you can do is you can have two outputs from the same controller. So you've got the same integrator. It impacts, in this case, it'll be impacting transcription of sodium, potassium channel proteins and synaptic channel proteins. And if you looked at the equations, they're basically essentially the same equation. And the rate of change is all based on the same difference between a set point, function of a set point, and the actual rate. And they both have the same fixed point where the firing rate is equal to the target. If you have two controllers, this is looking very complicated, but I've just kind of mirrored the same system. Now you have a target for one biochemical process and a target for a different one. And the target is going to be, the biological cell can't really say, oh, I want 3 hertz or something. What it can do is control different rates of reaction, and the reaction mechanisms are probably pretty much set, what sort of functions they are. But the odds are they're going to be different. If you've got two totally different molecular pathways, the reaction mechanisms are going to be different functions. There's going to be different concentrations of whether it's phosphatase or ubiquitin targeting different channels. So you're going to almost certainly get different targets, different target functions, and different set points. Now, if they were the same target function, you've got a problem. Because if we look now at the basic firing rate of a curve, again, going to a simple model, we've got these two mechanisms, okay? And essentially one mechanism is saying the rate must be its target rate. The other mechanism saying its rate must be the other target rate, which by chance are never going to be exactly the same. So this is having two thermostats in a room. One might be set at 72, one set at 68. And in a system unlike our houses where both heating and cooling can go on, you'll end up in the middle. One's cranking up the heating more and more, one's cranking up the AC even more and more, and which is clearly inefficient. And this is the problem of wind-up. If we did the null-Klein, here this is assuming a linear function, but whatever function you've got, if it's got to be different values, you're going to get contours, right? That are contours, if you look at maps, like I used to in the old days, they're never going to cross each other. Then if we plot the phase portrait, you just see then that you get this wind-up where both systems are working against each other, and until some maximums hit. And then you really don't have homeostasis, because you just, you might as well just send all the channels to their maximum wherever. Okay, so this was kind of a reason to think, well, why would you have two controllers, right? It's just causing problems. But you can solve this kind of stability problem. And this comes back to two features. One is that neurons, they're not like, what we'd ideally like our room temperature or our body temperature. The goal is not to get it to be a single value and fix there. Because if you're just trying to make things a single value, then you've got to hit the same value with both controllers. But we can then think of it as actually controlling a distribution. And one of the key issues is that the feedback is very slow compared to the timescale of fluctuations. So you can do this, a separation of timescales if you want. The other thing is the control mechanisms. I say it are nonlinear function. One could be linear, but the point is they're different functions. So in some sense, what you're doing is controlling either different moments, different quantiles, different aspects of a distribution, rather than either a single quantity or the same function of that distribution. And the simplest value, so this is kind of just what I'm saying. So we've got these two control mechanisms. They're controlling different aspects of a distribution. And just for intuition, the simplest way to think about it is, well, what if one controller were quadratic and the other were linear? Okay. Then between them, you've got one is controlling the mean square of whatever it's looking at, the other is controlling the mean. So between them, you've got both the mean and the variance. Okay. And so now you kind of see, and this is something in general, as well as the constraints, they both have to be kind of heading in the right direction on their own. You've also got another constraint, right? That variances can only be positive. If I'm trying to make the mean square less than the square of the mean, I'm going to be in trouble. So in this case, you've got an existence criterion, essentially. Variances can only be positive. And there also ends up being a stability criterion, which we'll look at later. You can get at from the Jacobian of the matrix. In this case, the way it's been set up, that if the threshold is just shifting kind of linearly, it's really better controlling the mean. Whereas the synapses, they're scaling up the inputs, they're more multiplicative. So that's really thinking of controlling the variance and the range. So in this case, it only works if it turns out the second derivative of the input scaling controller is greater than the second derivative of the threshold controller at the fixed point. So then we can kind of show that this does work, if you have those constraints. So on the left, we have a system with constraints, and this is showing, that the variance, what I've done, I should say, each of these sudden jumps at the top is a change in applied current. So this is the input coming into a cell. We can either double it, go back to the original, maybe 10 times the variance or 1/10 the variance. And both the means of the rates, let me just look at that. Yeah. And the variance of the firing rate comes back to its original. So we're kind of arriving at the original distribution, essentially. If you do things where we switch the targets, which is trying to force the variance to be negative, then you get wind up. This things they can almost work or appear to be working, but at some point you see that here the conductances are just going up and up and up. Okay, I mean here one is going down, one is going up, but the cell is silent by here. There's other types of controllers as well. We can put in, instead of quadratic and linear, we can use two Heaviside functions. And this is getting a bit more, exactly the biological functions we don't know, but Hill functions are used everywhere, which kind of have a maximum and start at zero. So Heaviside is kind of an extreme approximation of that. And then again, you can imagine, so then with a Heaviside function, what you're essentially controlling is what fraction of the time should the neuron be above a certain rate, okay? And again, there's going to be a constraint because here we have, 50% of the time, it should be above a rate of 1 hertz and 10% of the time above a rate of 15. That's okay. That's, that would make sense. And the controller works. We basically control the mean and the variance. But if you switch that and you say, oh, we've got a system that says 10% of the time the rate should be above 1 hertz, but 50% of the time above 15, well, of course, that's impossible. So that's kind of the same case as trying to ask for a negative variance. There's no way you can satisfy both controllers then. And you get the wind up again. So we cannot then understand, in these kind of simple cases. Of course, we wanted to show, that these are firing rate models where we can do the analysis and say when it's stable and show it. We want to do it, check a real circuit and also see if it can do something useful in the circuit. Would there be a reason for having two controllers, given you are adding additional constraints? So this is also work with Jonathan. And what we decided is, well, one thing was let's look at the mutual information. Neurons, of course, are doing more than just transferring their inputs, which is really what mutual information is asked. Like, how much do the output spikes tell you about the inputs? Well, then you could separate the neuron out of the system, join the input to the output, and you've done everything. But in general, whenever there is a step in information processing, loss of information means you're losing something. So maximizing mutual information was kind of a reasonable first step. We set up this nice adaptive exponential leaky integrate and fire model with a very strong adaptation. Basically, our goal was, and we'll look at this in the next task, we wanted to get kind of as linear a firing rate curve as we could. We got pretty close to a linear FI curve in a model. Mainly because in a minute we're going to be looking at the system with linear feedback, and we want to match the feedback to the inputs. So what we do then is we like jump the inputs through a whole bunch of different values, and in I think 100 millisecond bins count spikes to get an idea of the firing rate. And there's noise, of course, in the system. And without noise, everything, you can get perfect information in anything. And so then what we can do is like simulate models with different thresholds, different, so the threshold is here. This exponential, hopefully most of you have seen something like this in a neural model, but essentially the voltage gets high enough, this exponential becomes positive, it gives more input current, which is kind of like the sodium channels. And then as soon as you quickly get to a point of no return where the exponential is this dominating, this will kind of go off to infinity in finite time. We put a threshold, things depend very little on this threshold in this model. So we put a cutoff, right? Just because computationally it'd be with a limited time step you have to. Yeah, so it's really for computational reasons, but that's why the spikes are all sorts of different heights, because their rate of acceleration is becoming exponentially fast. So even with a small DT, you can get big jumps. I normally, when I plot these, this was Jonathan's, I would actually just force the spike to be the threshold if ever it goes above, and then they'll look nice and more realistic that way. So then we calculate the mutual information, and you can kind of see, here's a 2D plot. If the threshold is too high or the input is too low, basically it's not spiking at all. That's this region of 0. If it's spiking too crazily, too much, then you're getting very little difference based on the inputs. And you can kind of see there's a range, but the ideal range is if you go up here with a very high threshold and very high input, it's kind of binary. It's either spiking a ton or not at all, which is less information than if you can get a kind of graded firing, which is going on here. So there's a kind of optimal information. Of course, we kind of need to know the values to put it in to be optimal, which is another issue. But if you do do that, then the system works. You can start with the wrong threshold, wrong input conductances, and these will kind of move. These are trajectories that start way out here, and they'll move to this hotspot. One thing that I find interesting in this system, it's obviously not a surprise when you think in 2D, but for example, if you start over here, the goal is to have a very low conductance in the end. But initially the conductance goes up. And this is generally when you have two systems, there can be kind of an inherent oscillating aspect to it, where you might see one probably go up and down. And I know, like, sometimes they do the experiments and we'll see these things and they'll say, oh, a different mechanism's engaged. Because in biology, if you see a change in direction, the natural assumption is something different is happening. But a two-variable system, you can always see these kind of changes in direction. And the other thing, just showing that if you start from a random initial condition, then if you only have one mechanism, you could only be going in basically along one of these axes. Or if there's a combination, you could be going in a diagonal. But with two, you can get, you can basically optimize in 2D if you have two mechanisms. If you only have one mechanism, you're just seeing wherever you cut. It's kind of a 1D. Okay. And this system responds, and these are the sorts of experiments they might do. If you make the inputs way, way bigger, then here, I think we just scaled up by 10. Then this whole diagram is basically going to shift down by the Y scale by 10, and the system will just follow it. So you're getting similar responses. And if we keep the variance the same, but shift the mean, then it'll, again, drift in the right direction. Again, with this hit here, when both variables are changing, you often get a biphasic component to one of them. So we also wanted to show, well, is it, not just mutual information, but a kind of classic task. And so one that my first work was on this, and I kind of moved away from it, is tuning an integrator. So a lot of cognitive tasks, the idea of integration occurs if there's a continuous quantity that you want to hold in memory. So to tell the difference between, something's a bit brighter or less bright, or in the initial experiment, it was a monkey getting a vibration on a fingertip, and you see activities seem to maintain at different constant levels. And the whole idea of this is mathematical integration. If the input is 0, then the integration of that is just a constant, and you can maintain whatever constant. The other tasks that's used are a lot of these perceptual decision-making tasks where you get competing inputs, and there's an old theory that the optimal way to distinguish which is greater input when you have a signal and noise problem is to integrate both over time, because the noise increases at the square root, the signal, when you integrate it, goes up linearly. So, and there's some evidence for that in various decision-making tasks and so on. So this idea of, that the brain needs an integrator, and generally it requires fine-tuning, which is an issue. So the way that we would do models to produce integrators is you have neurons with a short time constant, but they get feedback. So their input depends on their firing rate. So here the input is just proportional to the rate. And we can kind of do this if we have a group of cells. If you have, say, 100 cells, then spikes are coming in pretty much continuously that you can actually talk about the rate being meaningful. Well, I guess we'll get into the equations of how it looks like integration in a minute. But the basic idea kind of visually is this, that you have feedback to the bunch of cells, which is proportional to their firing rate. And that feedback then is going to cross the input-output curve if there's no other input, say just at one point, okay, which would be a fixed point. To get an integrator, you kind of need to adjust the gradient of this and the threshold so that you get this situation where there's a line and things are never going to be perfect mathematically, unless they have to be exactly the same functions, but you need the two to follow each other. And the idea here is instead of one crossing, you're almost on top of each other for a large range of values. And essentially the difference in those curves is changing the time constant. So if you imagine one time constant, you kind of go from one curve to the other and back, then the number of times you do that to get to a fixed point is kind of telling you how long to get there. And in this case, if these are really, really close, you're going to be taking lots of little steps. So you need lots of like natural system time constants to move far when things are like this. And we kind of simulated this with a spiking network, and you get something that looks pretty decent as an integrator. So what we did is we kind of defined a factor, which is how good is the integrator based on there's kind of a decay. There's always, because nothing is perfect, a decay time constant. And you can kind of see here that there's really an unstable fixed point. This step is gradually going down after we have a stimulus input, it kicks it up. It's not 100% horizontal. This one is slightly going down, and these ones are slightly going up. So there are kind of two fixed points, but things are very, very slow in between. So we can look at the time constants and how many discrete steps you get and produce a factor that tells us how good an integrator it is. And then we can set the system up, so that it's an integrator. And again, the yellow is showing where it works really well. And then do things like decrease the mean drive to the cells, which is kind of like raising their threshold. Then the system moves and essentially reduces its threshold, goes down and hits the hotspot. In all these cases, it's hitting the new hotspot. Okay, this is kind of like moving it to the right. This is like moving everything to the left. And here we're killing 20% of the cells. So that's really going to reduce the feedbacks. You're only getting 0.8 of the original feedback per spike. So this is kind of like shifting the whole curve up because you need to increase the input conductance to compensate. So this system can compensate all these things in a spiking model. I realize I've been talking fairly fast. Are there any questions about that or any?
I came a little late because
Okay.
finding the room.
Sorry.
But anyway, with the homeostasis, of course, when things of it, you mentioned killing 20% of the cells. So is this a global property? That is to say, I mean, you might say a distributional property, because homeostatic, you need a lot of things.
Right.
To have a homeostatic.
So,
you need an ensemble.
yeah.
Having this.
So it could occur at the network level, and certainly different cells have different firing rates over a long period of time. At least 3 orders of magnitude, maybe 4. I mean, sometimes it's hard to see the lowest ones because, I think under 0.1, I think from 1/100th of a hertz, at least to 10 hertz plus average. And if you do perturbations, it seems like the same cells come back to the similar rankings. So it looks like the mechanisms are at the individual cell level. But that's, of course, if every cell, the mean body temperature of all of us in this room is going to be controlled if each of us is controlling our own. But they do have different set points, which speaks to the fact that these are like internal biological mechanisms. You're not going to have the same concentrations cell to cell in all these different reactants, and there's different sizes.
So you're saying homeostasis that's the result of an expectation of averaging, essentially?
No, I'm saying each cell has its own homeostatic set point, but they're different set points. Which maybe, I mean, temperature's not good because but maybe if you think of some other design, I mean, sleep, we all have like sleep homeostasis as well, right? But we probably get different amounts. I've heard of, Margaret Thatcher only got 4 hours. I don't know if anyone knows it, but someone in my family gets like 9.5 hours. It still feels she needs more. There's different ranges, right? But then as a population, you might say as a population, 7.5 is pretty much. I mean, it is interesting, and this is part of what we're thinking about in the next, why the cells have such a big range of differences. Which is not seen in motor control systems like in the Marder lab. They're pretty much, you want things to be a given a motor pattern generator needs to be firing at a certain rate pretty uniformly. You don't mean like our heart. We've all got ranges, but it's not 1,000-fold. Some of us have hearts 2,000 beats a minute, others 1 beat every 2 minutes. That's not going to work. So often in motor systems, things are a lot closer. But it is an interesting question. Why would you want in these sensory circuits such a huge range of firing rates? Because there's a sense that, well, not a sense, every spike requires energy. So if you're spiking too much, more than you need, you're using up more resources than you need. But also, just maintaining a cell in space takes a lot of energy. So if you're going to sit there, you better be doing something, right?
How are those set points encoded by these?
So this is what I'm saying. It's kind of the outcome of all these different reactions, which are going to depend on so many properties, like the cell size, different concentrations of all the other proteins involved in these pathways. So, like the rate of ubiquitination. What sets it exactly? I don't know. But there's a lot of factors. Some things are easy to know in a sense, that the amount of phosphatase, could be controlled by the cell, but it's also like the location of it, where it's happening within the cell. Is there enough phosphatase in that area to set the dephosphorylation rates? So how they're set, I don't know. I mean, the hand-waving answer is always evolution will make sure you're in the right range, but that might be that it may be if you don't need to be precise, that there could be a factor of 1,000, and so long as you're somewhere in there. I mean, so we can say what processes give rise to a set point, but I can't say how exactly the cell is controlling particular concentrations and geometries.
Can we experimentally change the set in vivo?
No one has. I mean, the one experiment where I know they have changed the step point, which seems homeostatic, is in C. elegans, where these tiny little worms, if they're on a gradient dish, a heated gradient dish, so one side is colder than the other. If they are raised at a given temperature, so if they're raised at 15, they'll move towards 15. If they're raised at 20, they'll move towards 20 Celsius. This is so, and this is in Piali's lab upstairs, where they will see a particular neuron, it will fire at 15 degrees and those rays at 15 to kind of adjust it where it's mode of motion, and fire at 20 if it's raised in 20. So internally in that cell, the set points are changed based on based on history, I guess. But again, how that occurs, I don't know. You know, so there's levels of knowing how it occurs.
A quick one more question. Is that something we can measure?
Well, you can see what a cell's long-term average firing rate is. In that sense, you do. And then when they do a perturbation and they see it come back, then you've measured. So you've measured the set point, if that's what you mean. Yeah.
If you're looking for a reason for having variability of set points in sensory cells, maybe it's just that sensory cells have to be able to respond to a wide range of stimuli which have different time scales, whereas motor cells,
The time scale set.
generating an output at essentially a fixed time scale.
Right. It might be, it's just then the cell responding on a very slow time scale. Just because codes can be combinatorial, why wouldn't that cell also do something else useful in spare time? But then anyway, kind of reminded of a funny story. The things I don't like things being on video because I don't want to big side stories. But my father was saying about, I think it was last time they moved house, and this one big guy, he was just standing there doing nothing, and he said, "Oh, don't tire yourself out, will you? Like, why is he doing nothing?" And then it came to moving the refrigerator, and the guy just strapped the refrigerator to his back and stomped up. He had this one job, and kind of was saving his energy for that one job. But I just don't know if neurons. I don't think I feel they could be doing something else in there in between. Yes, you were first.
Are set points kind of arbitrary given rates, like in way of their management of neuroplasticity and stuff like that?
I would not say they're arbitrary. There's something controlling them. But the point of the plasticity, part of what instigated this is a lot of plasticity mechanisms are inherently unstable. So the classic Hebbian neurons that fire together, wire together. If you're firing and then your input strengthen from excitatory cells, you're going to fire even more. And you get this positive feedback process. If you think of it being correlational, a positive correlation strengthens connections, well, that's going to enhance the correlation even more. So what you get is some cells going silent and others going to their max. So homeostasis is kind of to counteract that. But then what sets the homeostatic set point? Or you might say it's pushing things back a step, but in a lot of tasks, the range of the range of goal firing rates, 1 hertz to 20 hertz, you can still get a lot of stable operation of the circuit in that, and that can allow for a wide range of differences in inputs and still get stable circuit function. Whereas without that, everything will go to absolutely 0 or 100, maximum firing. So it might be, so what you find is that the control mechanism, you need to set those set points, but in our houses, right? In winter, it could go down to like below 32. I think the average high is like 32 in January and the lows are probably 20. So our houses will get to 25 Fahrenheit in the winter and 90 in the summer, right? But we can have set points, 55 to 75, and we would be okay. So you can get a range of set points where things are reasonable. Well, we don't know. So we don't know what sets the set points at the end of the day. But they are very wide ranging in visual cortex, layer 2-3 cells. You always have to have the provisos. Yeah.
I was wondering how much is the variability in the time to get to the status? For example, you have a G parameter at the threshold parameter. Will they come to the final stable point at the same time scale? Not necessarily, right?
No, it all depends on what the timescales of their processes are. So these are, in any model, these are parameters. And really, you have to think of the set point being one value. What here it looks like once you get reasonably close in good performance, then things are moving much more slowly. So what you find in a lot of these, moving to this general yellow band is very quick, and then moving along the band is slow. Because the differences from optimality aren't that big, you're close to the right variance and so on. I mean, the way this is set up as well, controlling the mean, that's just one of the control mechanisms and one variable. That will happen quickly because one is controlling mean, one is controlling mean squared, and they're both going to go pretty quickly. Now it's the difference that controls the variance, and that's a slower process. But that may not be how it works. You know, we just used mean and variance as a simple kind of example to show. I mean, I didn't mention this, but the way you can think about this working with the integrator is when you have a fixed point, as I showed before, where things are wrong, then you can kind of expand about the fixed point. Excuse me. And if you expand about the fixed point, you find you have an Ornstein-Uhlenbeck process. And the variance essentially goes down as the angle here gets wider. So if you're brought back very quickly, there's an effective time constant. And if that time constant is fast, you have low variance. If that time constant is slow, you have higher variance. And this is kind of like a little bit about what actually generates an integrator. Because an integrator is something with an infinite time constant, right? You change it and then it will stay there forever. So a lot of when we talk about is there an integrator or not, is kind of over what time scale. You can always integrate over milliseconds, you know, your inputs. But what you have is you have this natural time scale of the neuron, and then the effective time constant depends on the inputs and this derivative. And so as this approaches 1, you're getting towards an integrator. But notice, you know, to get 100 times the time constant, you need 1% fine tuning. This would need to be 0.99, right? And that's why, you know, when I was first doing it, it seemed like unlikely the neurons would be able to tune any of their, you know, connection properties or firing properties to 1%, because biology is much more noisy than that. But in this case, you can tune to that 1% or better by just saying, well, the firing rate has to be very variable with this sort of mean. Well, the only way you'll ever get there is as these lines get to fall on top of each other and you get to the 1%. I mean, in the case with just in this example, you end up either, you know, if this line is too shallow, you get a high fixed point. If it's too steep, which means very little feedback in this sense, you'll get a very low rate fixed point. So even if your fixed point rate is somewhere along this region, it's kind of forcing them to come quite tightly together. But the point is here that, you know, in this case, the synaptic conductance is changing the effective time constant and is via the variance because it's in the variance of the firing. Okay, so I might just talk about some of the problems that arose. And essentially what happened was this. Gina, she came to my office and said, you know, so it looked for a number of years that, you know, a certain kinase was involved in both mechanisms of homeostasis. So maybe it's really only one mechanism and we don't have to worry about this. But more recently in the Turigiano lab, they've kind of combined different experiments. And what they know is that, so here IAMPA, these are the synaptic conductances, the connection strengths. They depend on calcium entry into the cell body through particular calcium channels in the cell body. Whereas, and you can do this by blocking channels or chelating calcium in the soma. Whereas the intrinsic excitability, which shifts the firing rate curve, that depends on calcium entry through NMDA receptors, which are also kind of like AMPA receptors involved in the connections. So now things are looking from the outside even more weird. It's not like just having 2 thermostats in a room, but if you have a split level house, it's kind of like having the upstairs AC and heating controlled by a downstairs thermostat, and a downstairs AC and heating controlled by an upstairs and thermostat. And then it's like, how on earth can that happen? So, you know, we haven't solved this yet. Some of the ideas are, you know, that back propagating action potentials are important. So if you increase sodium conductance, then you'll get more spikes, which could send back a rise in voltage to the voltage. And then these channels, the NMDA channels are actually voltage dependent, so let more calcium in up there. But it's hard to separate the two mechanisms. So first of all, we have a lot of unknowns. So we have these conductances, they depend, one of them depends on a function of the NMDA calcium, the other depends on some function of the somatic calcium. But we can generally just like describe these two systems very generally. Find the Jacobian, and the question is, when is the system stable? Well, when both eigenvalues are negative. And you kind of see, for the trace to be negative requires these, this sum to be greater than 0. And the trace is basically like the diagonal is essentially if each mechanism is heading in the right direction, it will have a negative derivative. Okay, so if temperature goes up, then something should be making d by dt negative. The other one is on other conditions on the determinant. And again, it's somewhat intuitive, but if you look at the product of each mechanism's kind of effect on itself, it had better be greater than how it impacts the other. So this would be like the thermostats, that somehow the downstairs thermostats impact on the downstairs temperature, multiplied by the upstairs thermostats impact on the upstairs temperature, had better be greater than the product of the opposite way around, which if you put them in the opposite positions, naturally is not going to happen. But what we decided to do is I did like numerical derivatives to kind of estimate these Jacobians. So you can just have the cell, firing away, measure the whole distribution of calcium and say what happens to it if you raise sodium conductance, what happens to you if you raise synaptic inputs, and you can get these shifts in the distribution. And the nice thing about this is now, given the change in distribution, I can try every function under the sun and every pair of functions. So I have kind of like, not just mean variance, I have like every, different quantiles, and then see how those quantiles change the calcium, and then would change these functions and calculate the derivatives. So in that sense, like if you have 13 different functions for this, 13 different functions for there, I can try like 169 mechanisms, having only really done 3 simulations, base calcium, slightly raise sodium, slightly raise the synaptic inputs. And these simulations, just a practical point, these are slow because when you have all these channels, you need very, very small time steps because, a typical Hodgkin-Huxley model, you might need 10 microseconds time steps. You can get away with more if you use Runge-Cutter 4. But the homeostasis is on hours. So it just means a very, very slow simulation. So being able to, cover loads of possible options very, very quickly helps. So, sometimes we can get things working. So we did this one where we kind of blocked the NMDA. And this is the case where the intrinsic, the sodium potassium channels are meant to change and not the synaptic conductance. And what happens is you get an initial change, you're bound to get some change. And this is what I always find in the other property, the ones they see that don't change. But if things are right, it can be it can rebound and go back down. So you don't see anything. And this is what happens here. But it relies on a couple of things. We kind of need the mechanism that is observed to have been affected to kind of max out at just the right level so you don't really see a change in the other one to match the experiments. The other thing we find, and this is just showing, I should say that this works reasonably well in different circumstances, that we can reduce input. We can start from different initial conditions. So you could get the range of rates and it can still be stable. But the problem is this, that those control points is still very sensitive. So here I just, maybe 24.5 micromolar was ideal and everything worked fine. If I made it 25 or 24, I got wind up. So things are like very, very subtle. And this then is just a real issue, right? Because the whole point of homeostasis is you don't need to fine tune things. If we need to fine tune our set points, then we've lost all the benefit of homeostasis. So, that's kind of where we are. There are benefits to a dual system, but there's always, I think this wind up is always lurking in the background. It can easily happen if things are not quite aligned. We get these distinctive hallmarks. You know, that where you might find there's a dual system, you always kind of have to look at where the two pathways branch, and then is the control mechanism after the branching or before, because there's always going to be a lot of there can easily be a lot of steps that are on the same, that are contributing to both mechanisms, but it's just where the integration happens matters. But you can also find them happening in opposing manners. And one thing these are hard experiments, but if you could block one mechanism, you might find the other was occluded, and does appear in some experiments. And just kind of, what I've started doing is working on these 5 compartment models, in part because one of the things I mentioned is, the sodium spikes, you want to have a big impact on NMDA input. And in particular, that for that NMDA mechanism, that if you don't have enough NMDA input, you have more sodium spikes produced in the soma, and that will now increase your NMDA input. Well, there's also sodium channels in the dendrites. So by putting in this intermediate dendritic step, it can basically then, if you enhance sodium conductance, allow spikes to propagate, back to the distal dendrites and impact the NMDA channels more. So it's kind of what I'm trying to do is, from those different components of the Jacobian, enhance the diagonal ones and reduce the off-diagonal ones. And having the basal dendrites, the other thing I was noticing is that whenever the soma spiked is because you already had high input through the channels. So by having different branches, then the somatic spikes are less correlated with any one specific input. So we're kind of like trying to decorrelate the off-diagonals and correlate the diagonals to make things more stable. But we have some other ideas as well. Like there might be, rather than this be homeostasis that's always on, might be actually a little more like our houses, where you could imagine setting the heating only comes on if you get below 60, and the cooling only comes on if you get above 75. And then you've got a range where maybe things can be robust. That's another aspect we're looking at. Anyway, thank you for your attention, and sorry if I've spoken a little quickly. Hi.
But I need to think a little bit more carefully about some. For example, like, why would you imagine that two controller? Isn't it plausible that two controllers have a similar set point? Because, for example, if the cell has an identity, I want to fire at 1 hertz, and I have a bunch of different knobs that they get my guy to fire at 1 hertz. Well, then wouldn't both of those controllers have the same target?
But the cell doesn't know it wants to fire.
So
The cell's just responding to inputs, biochemical pathways. So somehow it's got to get this reaction mechanism in this part of the cell to know the same target as this reaction mechanism in a different part of the cell. Now, if the mechanisms are kind of, if there's just one mechanism, but calcium varies across cells, cell regions. I mean,
sure, okay.
But even then, that a given level of calcium, unless the Hill functions are exactly the same, it's going to produce different reaction rates for different kinases, different phosphatases. So unless it's exactly the same,
wouldn't you want it to be flipped? Because the cell ultimately, like in these motor patterns, right? They'll fire at some fixed rate, right? Or
right,
within some rate. Like whatever the outcome of the variations of calcium are, ultimately, the output is not something that's conflicting with itself.
So in the motor system that you're talking about, which is very different, in that you do want pretty regular.
Right, so that's what you were saying.
Yeah, then it appears there's certainly evidence from these correlations in channel conductances that there might be one controller with multiple outputs. So then it is got one set point. And if you're upregulating one channel, you're like upregulated a similar one or downregulated an opposing one. So the changes are correlated because they come from one controller. In this case, if you have two controllers, the two mechanisms, as soon as there are two involved, they're not going to be identical, in which case you're going to find You've got issues.
In principle, like the network, why wouldn't the analogy extend? I mean, I understand what
the analogy. I know, the key thing is, if you have an integration step, that's the control. Is the output.
I'm talking about the target stuff. Like in the end, like if I want a network to fire at on average at 1 hertz with a variance of like 1 hertz, wouldn't all of my controllers want to target that identity?
Well, one controller can only address one property, one function of the calcium distribution. So if you think of the mean and the variance, right? The one controlling the variance also has to know the mean and make sure that its goal is like 0.01 above it or something. Like if one is 1 and one is one variance between 1, I'm not sure what it is. Mean squared would be 1.1. But how would you, and the nice thing about previous controls with one controller is you can set a mean at 5, a mean at 7, a mean at 10. The neuron's going to be fine, and you'll see, oh, this cell's firing a bit more, the other cell's firing a bit less, but it can still function. If you've got to set, oh, this one has to have a goal of 10, this one about to have a goal of 10.5, it's going to miss. There's nothing in the book generally. So if you say, if you look at some of the things we're looking at and say that, well, it is controlled by a different function, and that function has a big range, then you have a lot more room, a lot more tolerance. But it can still get lined up. If it were to go below 10, you'd have a problem. But you could set the mean square goal at 15, at 20, at 12, and you're okay. But if the mean goal is 10, as soon as the mean square goal goes below 10 squared, you go.
I was wondering, is there going to be a confusion of homeostasis with two integrators? I was wondering if there's a possibility of something like limits at first, or that's never almost that.
No, I think you can. We saw something interesting, and there is actually, it's kind of a connection which I've not had time to really follow up. But in the hippocampus, which is kind of the scene for episodic memories, a key aspect of episodic memories is connecting events that occur at a similar time. So I'm looking at the person from Chantal's lab who works on this. And it is observed that if you have a new experience for an animal, that you can predict which cells are involved in it by their excitability at the time, just before the time of the experience. So you can kind of, if there's a new arm of a maze that has, what they'll do is they'll give specific features to it so it knows it's somewhere different. The place cells there will be the cells that haven't had higher activity beforehand. But then if you wait a few hours later, different cells will have higher activity. So one thing that has come out of some of these models is the activity, there being an oscillation in these homeostatic mechanisms, which will give rise to that. And essentially, I was kind of seeing it as a bit of a bug, but what happens is cells become a little bit silent, and then the mechanisms start making them fire more and more and more, and then they start bursting because they're firing too much. So if you've got a cell that can burst, then it can never quite hit, that 1 hertz firing rate. So it'll start firing at 10 hertz, and then the mechanisms will kick it back down. So you can get this kind of feedback, which is basically a limit. But they may exist in hippocampus, because I don't know how regular an oscillation is, but certainly cells are going up and down over hours, timescale of hours, which is useful for episodic memory in the excitability. Yeah, I realize if anyone else needs to go, I shouldn't, because I know we're a little over. But yeah.
Is the wind up biologically? I know it is a bad thing, but is it biologically, can we explain some neurological disease and like seizures? Does it happen, or no, it's just a mathematical thing?
So, generally there are two, and for those online who know about this, I apologize. But epilepsy is often considered, there's a lot of synchronization, but generally if neurons are too excitable, that's going to happen. And depression is also associated with lower than typical activity in neurons in the brain. So you can get these issues where you're going one way or another. I just don't think wind up would happen because wind up is so bad. I know always other things are going to come in to avoid it. But we did have one idea, which I'm not sure how easily we can test, but this 3 or 4 more orders of magnitude range in set point. So one of the mechanisms you may know about the critical period. So a lot of sensory systems, they're very adaptable, and there are a lot of, they will change a lot in their connection strengths and so on during certain windows of time. And I think there's like a week or so for V1 in the mice. That this homeostatic plasticity, one of the mechanisms is observed only to be occurring during that window. So all these changes in connections are really destabilizing, so that's when you need it the most. But there might be an idea there that it is actually causing the neurons to spread out in an unstable manner, but then because it stops, you're left with this whole many orders of magnitude range. So it could actually be useful for that. That's one hypothesis. If it was on forever, it would be a problem. But
Yeah.
you can have if you have a finite period of instability, things spread out, and now you've got your huge range of firing.
Cool. Thank you very much.
Oh, glad you enjoyed it.